You can bring a page of notes again, like 8.5 by 11-inch paper. I would like it handwritten, one-sided. You can draw pictures if you want to, or take a picture from the, like a picture from the slides if you want to, just not a whole slide. But any photos should also be pasted into the paper as well. So just this size of paper, okay? But definitely bring notes because that will help you. I wanted to just show you the learning outcomes for the course, and what we've already done is in green, and what exam 2 is about is in yellow. So metabolism, and that's the aerobic and anaerobic respiration. And then what we did today was analyzing the microbial agents and developing recommendations for use of them. And then we have done the growth and growth control labs. So those are what you're going to be tested on. That's chapters 5, 6, 7, really. And so the lab content is a lot of chapter 6 and 7 material. You won't be actually setting up experiments and doing the experiments, but you'll be observing test tubes or plates, things like that. So let's go over a few of those. So remember, this is the temperature ones. So the cardinal temperature term for this, if we look at the temperature ranges here, I will give you this chart.
Oh, cool.
Okay, so you don't have to put that on your notes.
Oh, thank you.
Yeah, but you should be able to know how to use the chart, right? So here we have growth in 20 degrees and 30 degrees. So what's one word that we would use to describe this organism?
The 30 degree is mesophilia and the 20 degree will be
Okay. So you have to use one term.
Oh.
All of the growth is considered, has to be under the curve. So 20 and 30, the only curve that is under, that those are under both of the, or both of those temperatures are under the curve, is the mesophile. So the 20 is touching the psychrophile, but psychrophiles cannot grow at 30. And so it can't be psychrophile. It is just the mesophile. So one word.
So the measurement is where they connect, not the top. The top will be.
So it's between, this is their range of growth.
Okay.
So it can grow, if it grows at 20, 30, 40 even, if it was growing at 40, it would still be a mesophile.
Like it goes the best.
But if it grew at 50, then it shouldn't have grown at 20. Because that's outside of the range of the thermophile, right? Okay, so then let's look at this one here. It's growing at 50 and 60.
That's thermophile.
Thermophile.
Yep. So 50 and 60 is under that red curve. So thermophile. So the peak means optimal growth.
Okay, okay, yeah.
Yep. But the edges are the range of growth.
Okay.
So for temperature, we're looking at cardinal temperature terms, we're looking at range of growth. All right. I also showed you this when we talked about temperature. And so these two tubes have the same organism. One of the tubes, the organism's yellow, kind of yellowish. The other tube has red in it. They're incubated at different temperatures. So what does this tell us?
Does the red one need oxygen and that's why it's up?
Nope, nothing to do with oxygen.
No? Oh, no.
So what does it have to do with? It has to do with temperature, right? So temperature, there's different temperatures which cause different colors. So sometimes pigment production is temperature dependent. That's what that tells us. So if we have these two tubes, two different temperatures. One comes out red growth, the other a different color growth, yellow growth. That means that temperature was determining pigment production.
Temperature, you said again.
Is determining pigment production
Okay.
or color production.
I have a question about that.
Yeah.
Is there gonna be anything else like on the test that it would determine? Like, could it be something else like not pigment or like growth in general, or would it just be pigment?
For this setup, it would, if it's incubated at two different temperatures, it's just the pigment that it's showing us. Yeah.
Thank you.
Okay, so do you remember, I did talk about that, when I was talking about putting like an organism as a bioindicator in the incubator and knowing if the incubator turned off overnight because it made a red color. Remember that? Okay, so salt. We're gonna describe this organism in terms of osmolarity. So, there are some terms associated with salt lovers. So, the growth is the cloudy ones, which are 1% and 2%. So, what do we call this organism? Non-halophile. Good job. So it's a non-halophile because it doesn't grow at 3% and 4% salt. So what happened with 3% and 4% salt? What happened to the organism? Plasmolysis. Yep, it died. So plasmolysis occurred at the 3% and 4%. And so that's why there's no growth there, because it's a non-halophile.
And when does plasmolysis occur?
What did you ask?
At 3 and 4?
Yeah, 3 and 4% salt. When it cannot withstand that salty conditions,
Okay.
plasmolysis occurs.
And then we could have like different results in the test. Like this is just a.
Yep, could have different results. This is just an example. So if it was growing at the 3% and 4%, instead, we would call it a halophile.
So at 3 and 4, it could not handle the salt. Is that what you said? So it's plasmolysis.
Yeah, that's what happened in this case, when there was no growth at 3 and 4%.
Are we using the word extreme?
Extreme halophile would be if we're talking about
15 to 10
15%.
to 25.
Yep.
After 25, they will die, right?
Yeah. Okay, so for pH, pH 4 and pH 6 has the cloudiness for this one. So what do we call this organism? So what is pH 4? Is it acid, neutral, or alkaline?
Acid start from
pH 4 is acid, right? 0 to 7, 7 to 14. And then you move away from neutral from 7 toward 14 is alkaline, toward 0 is acid.
So pH 6 is.
pH 6 is technically slightly acidic. So what do we call this organism then?
acidic.
It's a lover of acid. Acidophile.
Acidophile. What did it say?
So,
Acidophile.
So those are, you may have to, or you will have to interpret some test tubes and tell me the right term, okay? And they'll be like this, they'll be straightforward, so it'll just be within that range of acid or neutral 6.5 to 8.5, or base is 8.5 and up.
So we'll just wanna make sure we have the ranges.
Yeah.
So we consider all of the test tubes too?
Yep, you look at all the test tubes and you see where it's cloudy.
And then go off of the, if there's two, then go off of that range or whatever.
Yes.
Yeah. Okay.
So if there's two cloudy ones, they're both in acid range, so it's an acidophile.
Okay.
The range is for the acetyl.
Acidophile is from 0 to 6.5. And then 6.5 to 8.5 is the neutrophiles. And then the 8.5 to 14 is the alkalophile.
How come it didn't grow in the pH 2 then? If it's
Because
on the range?
it just prefers slightly less acidic than that, yeah. It was just too acidic for that one.
Just to make sure we've got the pH ranges.
It likes it acidic, but not too acidic. Okay, so I know this is a lot of words here, but this is just to let you know all the parts that need to be described. If I ask you to describe an experiment and interpret the results. So there will be a question about either pH or salt, okay? And so you'll be needing to, maybe I say, for example, on this one, if you're trying to determine if an organism is an extreme halophile. So you are going to set up the tubes with the different salt solutions. Make sure you have testing for extreme halophiles, so testing tubes that are 15, 20% up there, not over 25, you don't really need that. And then some that are less, just to make sure it's not osmotolerant, right? Because osmotolerant's all of them. And so you explain that you set up these tubes, you inoculate the tubes, you incubate them, you come back later, it doesn't even matter if you say 24 or 48 hours, whatever. Come back later and you look for turbidity or cloudiness, and you find it cloudy in the 15 and 20% salt, and so you can make the conclusion that it is an extreme halophile.
And we'll determine those, so after we come back from the,
You
or-
will not be setting up the experiment.
Oh.
You will be describing it.
Oh, okay.
Yep.
Got it.
Just the salt one?
Might be salt, might be pH,
Okay.
but it's the same sort of setup,
Gotcha.
right? The tubes, the inoculation, the incubation, the cloudiness,
But,
and the results.
so we will set up the tube though and stuff?
No, you
No.
will describe
Okay.
it.
No, you
Oh, I'm
just
so
do
sorry.
like
Okay.
this, you do just a small like.
All right.
this.
Sorry.
You
It's
can
okay.
also just make a drawing. So if you're starting to run out of time, you can just draw the tubes, label them, the different percentages of salt for this example, and put the cloudiness in the right ones, and then just a little bit of description. So you don't have to do a whole essay. It can just be like that, and I'll know like, okay, they know they're supposed to do the tubes. They know they're gonna put a note, you put a little note saying, I'd incubate the tubes, I'd come back later, this is what the results would look like. And now I know it's an extreme halophile. So it's okay if you use diagrams for that. Labeled pictures are fine. Makes it go a little faster to explain. Some people don't like to draw though.
I do.
Okay? So again, not setting it up, just describing it and interpreting the results. Okay, so here's oxygen. When you're testing for oxygen, what is the term used to test for oxygen? Aerotolerance.
Aerotolerance.
Yep, aerotolerance. And there's 3 methods we used, and we can use abbreviations, it's fine. So what was the solid one, do you remember? Has 3 letters. BHI, and then the liquid was FTM, and then we also use this thing, the anaerobic jar. Anaerobic jar. Okay, so if I were to ask you to use 2 words to describe this tube here, the results in this tube, we have growth here, and then there's some growth down here. I know it's kind of hard to see that on this screen, but you have growth here and some growth down here. So what are 2 words to use to describe that organism? Close. Facultative. And then the second term? Anaerobe. Yep. Facultative anaerobe for this one, where there's growth up here and then some growth down in here. Facultative anaerobe. And then this one here, just growth up at the top.
Obligated arrow.
Yep, obligate
Obligate
aerobe.
Obligate?
Obligate aerobe. So make sure you remember that facultative goes with anaerobe. Okay, so what is it that we're testing again for using this apparatus? One word.
My
Aerotolerance. Aerotolerance
Aerotolerance is the apparatus.
is what you're testing. The apparatus of it is an anaerobic jar. And what grows well in an anaerobic jar?
Oxygen.
Anaerobes grow well in
Oh, yeah.
anaerobic jars. The anaerobe grows well in an anaerobic jar. Okay, this is just quick recall. What is the name of the test we were looking at for antibiotic susceptibility? Yep, good. Kirby-Bauer test. And the name of the agar?
The Hudson, or Hinton, Holton?
Yeah, it has Hinton in it. Good. Mueller-Hinton.
Mueller-Hinton.
And which one was more susceptible, gram-positive or gram-negative? Right, gram-positive was easier to kill. Oh, gram-positive. Yep. So susceptible versus resistant. So susceptible, we should go ahead and prescribe it. Resistant, don't prescribe it. And we tell that by looking at the zone of inhibition.
Resistant.
So susceptible, you're gonna prescribe it. Resistant, don't prescribe it. And you tell it by the zone of inhibition.
Yeah. What was the, which I'll study anyway, but the Hinton-Mueller?
Mueller-Hinton.
Mueller-Hinton, and it's
Yeah.
the Kirby-Bauer test, not
Correct.
Mueller. Sorry, I told you
Yeah.
Kirby-Bauer.
Oh.
Or Kirby.
Wait, I have a question about that one. So, which is more susceptible? Would we decide by something there, or is always going to be like?
Gram-positive is just in general more susceptible to antimicrobials. And you saw that on your plates. Okay, so this one, here we have the P, T, and C all have 0 millimeters. So what does that mean about P, T, and C? Penicillin, tetracycline, and chloramphenicol. Should we prescribe that one? So the ones with 0 millimeters,
We shouldn't.
yeah, that means they're resistant. It's resistant to those. If we were trying to choose between gentamicin and levofloxacin, we have gentamicin has 15 millimeters, which is just above the susceptible value. And then levofloxacin is quite a bit bigger than the susceptible or sensitive value. So probably levofloxacin would be the best one to prescribe in this case, because it has a much bigger zone of inhibition than is needed.
So meaning it'll kill it. Good.
It'll kill it.
Yeah.
Yep. Okay. All right, so, if you're looking at these two plates, which one do you think is gram-positive, A or B?
A.
The one that is more susceptible is gram-positive, right? So the one that the antimicrobials kill more easily is B, and that would be gram-positive. The ones that are it's killing it more easily. Okay, the one that works for both, probably bleach in this case. Do you understand why I said B would be the one that is probably gram-positive? No? Because the zones of inhibition are bigger and gram-positives are more susceptible or sensitive to cleansers or antimicrobials. Does that make sense?
You said what is more sensitive? The
Gram-positive,
gram-positive is more
more sensitive to antimicrobials.
to antimicrobials.
Yes. Okay, what organisms are easiest to kill with UV light? Is it gram-positive, gram-negative, or endospore formers?
Gram-positive.
Gram-positive, yep. So just get it into your head, gram-positive or easier to kill? Gram-positive or easier to kill? Easier to kill with UV light, easier to kill with cleansers, with antibiotics, they're just easier to kill. They don't have that extra membrane, that's why. If we have the choice of gram-positive, gram-negative, endospore former, which one is the hardest to kill?
Endospore.
Endospore former, yeah. And what, the ones that are killed, how did they get killed? They got killed by damaging the DNA. That's all I really need to know. It's these kinks, these thymine dimer kinks that cause the DNA to be damaged. It happens more frequently than the cell can actually repair it, and so then it just dies.
So they get killed by the DNA dying. Yeah.
By the DNA being
killed.
destroyed,
Okay.
damaged. And that also will disable viruses too that have DNA in their replication cycle at all. What's ubiquity mean? Found everywhere, yep. Okay, so for chapter 6 material, it's mainly based on the lab experiences that you've had and the stuff that I've gone over.
Girls everywhere. Oh, phones
But the
everywhere.
only thing that wasn't really talked too much about in lab was this growth curve, and we'll be using this information during your unknown identification project. But knowing the different stages is all you need to do, be able to label the different parts of the growth curve. So the first phase is the lag phase, where you've just inoculated something and you need it to grow, so you put it in the incubator.
So that will be the new introduction to adapting to the environment?
The lag phase? Yeah, you just introduced the organism.
That's number one, right?
Yeah, number
Okay.
one. You just introduced the organism, so it's just been inoculated. Number two, the log phase, they're just increasing in numbers, they're replicating, so you put that in the incubator, that's for the incubator as well. In the stationary phase, they're at maximum population size, and so the death and cell division happen about the same rate. Usually you want to stop growth at that point, so you put it in the refrigerator. And then if the organisms are kind of been growing on that media for a while, then they start to go into the death phase, because their wastes are building up, nutrients are being depleted, and so you put it in the biohazard bin.
You said they're at their maximum what size, or maximum?
Population size,
Population.
the stationary phase.
Thank you.
So will be the organism dead one? So nutrition use waste is increased.
So I think that's the only chapter 6 material that wasn't really covered in lab. And then the chapter 7 material was all about antimicrobials. So there's a few things here. There's both lecture material and lab material. The lecture material is kind of this stuff here. So sterilizing versus sanitizing. So remember, sterilizing is killing all living things,
All microbial life.
all life really, versus sanitizing, you are just lowering microbial counts to safe levels. So if you think of like hand sanitizer, it's not 100% effective, right? 99.9 or whatever. And so because there's that 0.1, it's still lowering them to a safe level. Just not killing everything. You don't have hand sterilizer, hand sanitizer. When you flame the loop, that is dry heat sterilization.
That's sterilized, is that what you said? Yeah.
Dry heat
Yeah.
sterilization, yep. And then the machine used in hospitals is called what?
A sterilizer? Or
No.
sanitizer?
Autoclave.
Mm.
It's the autoclave.
And that's what I stopped yesterday.
Hospital, what they use, is that what you said, to
Autoclave.
sterilize?
Sterilizing,
Autoclave.
sterilization machine in the hospital. Yeah.
Autoclave?
Autoclave.
Autoclave.
AUTOCLAVE. And then there's a variety of water cleaning methods that I want you to look over. So make sure you're looking that over. So some of the chemical methods are like iodine. Sometimes you'll clean water for drinking if you're hiking. You do iodine tablets. Chlorine is used usually for swimming pools and sometimes spas. I have another one in my head. Bromine is another one used for like hot tubs usually. And then for another some physical controls with water, like filtration is another one. So you know, like those life straws or filtration machines. Ionization or UV light is also another kind of physical method of control. Probably the best one for drinking water is boiling. sometimes called pasteurization, but boiling is sufficient. So if you're trying to get water clean for drinking, that's probably the best way. Or bathing, but that's just a whole lot of water you're trying to clean if you're bathing with it.
You said UV light for water, what you said?
UV light can be
Yeah.
used for water, that's like ionizing radiation. Sometimes they have those reverse osmosis, that's like filtration. Okay, then for chapter 5, you want to make sure you study the study guide for chapter 5. The enzyme part mainly comes from the enzyme worksheet that's on Canvas, and I talk about in the lecture material. Some basic stuff from this chapter. Oxidation is loss of hydrogen atoms, so loss of electrons in the form of hydrogen atoms. I don't know if you've seen the acronym OIL RIG, but oxidation is loss, reduction is gain. Those are good like mnemonic to remember what oxidation and reduction is. So if you're losing something, you're losing hydrogen atoms, you're also breaking. So oxidation is a breaking type of reaction, lysis reaction. So glycolysis is oxidation of glucose. Pyruvate gets oxidized if the oxygen is present, so that gets broken. Lots of oxidation happening. Reduction is the building or the gaining of hydrogen atoms. So if it goes from NAD plus to NADH, then its reduction happened, right? It gained hydrogen atoms. And the focus will be on oxidative phosphorylation rather than the other two. I talked about substrate level and photophosphorylation, but oxidative phosphorylation is what you want to focus on, which is in the electron transport chain using oxygen and the products of all those redox reactions. So there's only 2 non-respiration related, or I should say non-respiration, non-fermentation related questions. Photosynthesis, so the photo part is the light reaction, the synthesis part is the Calvin cycle. That's basically it. I didn't go too deep into photosynthesis. And then there is some anoxygenic photosynthesis, meaning that oxygen doesn't get produced. And so photosynthesis takes carbon dioxide and water, and then you get glucose and oxygen. So that's oxygenic, right? It makes oxygen. If you don't have water, then you can use hydrogen sulfide, H2S, instead of H2O, and you get sulfur out of it. So you do anoxygenic photosynthesis without water using H2S instead of H2O. Kind of cool they can do that. Okay, so the enzymes, check out that practice problem worksheet because it gets at the kind of basics of what you need. When you're interpreting the graphs, if I ask for optimal, I'm talking about the peak here. So if I ask you what is the optimal temperature and pH for this for this enzyme, then you would say somewhere around
38, 37.
38, 37, 40-ish, whatever, right around there. And here you would say pH 8. Okay, so being able to interpret those graphs that are related to enzyme kinetics. And then here's 2 other ones to make sure you can interpret as well. So for this one here, we have 2 enzymes represented. You have one that works well in the acidic conditions, And another one that works well in alkaline or basic conditions. So here's another test to make sure you know that what pH is. So if we have both of these mixed together, they will work, both of them will work in this range of pH 4 and 6. But if the solution becomes more acidic, then your gastric protease will work better than the intestinal protease. Or if the pH is adjusted to be higher, like a pH 10, or let's say pH 8, then the gastric protease will stop working and the intestinal protease will be working well. So that's the interpretation of that graph. I will ask you questions not exactly the same as what's on the enzyme worksheet, but very similar. And then for this one, there's 4 different enzymes. For this enzyme question with the graphs, just look at optimal. Don't worry about how wide of a range. Okay, just think about the optimal rate of reaction.
Can you
And then
repeat that
pH.
one more time? Sorry, you said,
Yeah, focus on the optimal rate of reaction. Don't worry about the range
okay.
of the as much. Okay, so here's some more differences in the metabolic processes between eukaryotes and prokaryotes. Locations. So for prokaryotes, glycolysis, fermentation, pyruvate oxidation, citric acid cycle, they all happen in the cytoplasm. That's the only option, really, because they don't have all the other organelles.
Those all happen in the cytoplasm.
All the glycolysis, fermentation, pyruvate oxidation, and the Krebs cycle. The electron transport chain in prokaryotes is on the cell membrane.
Can you rephrase yourself, please? I'm sorry.
Sorry?
Can you rephrase yourself?
Yeah, so the electron transport chain for prokaryotes is on the cell membrane, or the plasma membrane. And chemiosmosis occurs from the periplasmic space across the cell membrane. So remember, chemiosmosis is when the hydrogen ions come back through ATP synthase. So they go from the periplasmic space, which is outside the cell membrane, across the cell membrane into the cytoplasm. For eukaryotes, glycolysis, fermentation, pyruvate oxidation mostly happens in the cytoplasm. So the first 3 processes happen in the cytoplasm. Sometimes pyruvate oxidation can occur across the mitochondrial membrane. Just depends on the organism. But pyruvate oxidation for this is fine if you have it in your head as in the cytoplasm.
And the chemiomyosis is where we find the high proton concentration?
Yes. The Krebs cycle or the citric acid cycle in eukaryotes happens in the matrix of the mitochondria.
happens in the mitochondria.
Mitochondria matrix.
For eukaryotes.
For the eukaryotes. Prokaryotes do not have mitochondria. The electron transport chain happens on the inner mitochondrial membrane. And chemiosmosis occurs between the intermembrane space across the inner mitochondrial membrane into the matrix. So it's just in the mitochondria where chemiosmosis for eukaryotes. The intermembrane space across the inner mitochondrial membrane is where chemiosmosis happens for eukaryotes.
Membrane. It speaks of the
Space
mito-
of mitochondria.
What was the last word? Mitochondrial membrane?
Membrane.
Yeah,
Membrane.
membrane. Okay. That membrane.
Okay, so for prokaryotes, we've got cytoplasm, 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 then plasma membrane, and then periplasmic space across the cell membrane, across the plasma membrane. For location in eukaryotes, we've got cytoplasm, 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 mitochondrial matrix, inner membrane of the mitochondria, and then we have chemiosmosis is intermembrane space across the inner mitochondrial membrane. So there's some metabolic diseases and some medications that are related to mitochondrial function, and some medications that have actions related to the mitochondrial function. So when you're in your pharmacology classes, you're gonna apply that information of like where this all is happening when you're thinking about how drugs are being used in a body. So when you're in like nursing school or dental hygiene school and you take pharmacology, they'll expect that you know what intermitochondrial membrane is and that ATP production happens there, and it's important to have that that gradient of protons going from high concentration to low concentration, because there are some like arsenic poisoning. If you poke holes in the what it does is poke holes in the inner mitochondrial membrane, and so then the protons just leak back into the matrix of the mitochondria, and you don't get very much energy being made and not enough that's sustainable for life. And so that's how arsenic poisoning works. And so to reverse that, you have to have something that pumps the arsenic out of those mitochondria, so that you can get it quick enough to repair that and protect the cells from that. So there's some medications that act at the mitochondrial level, and so that's why they want you to know that before you go to nursing school or dental hygiene school. So this is stuff you'll need to know later. And that's why we talk about it in every single one of your classes, right? Almost all of them. Okay, so glycolysis, I know I went into deep detail of glycolysis in the lecture notes. Really, all I need you to know is that glycolysis glucose gets oxidized to pyruvate, NAD, and ATP. So you don't have to know all the deep detail that I covered in lecture. Just the summary of you get glucose in, ATP in, you get pyruvate out, ATP out, NADH out. I don't know why this picture doesn't have that, sorry.
You said glucose gets oxidized to pyruvate, right?
Yep.
Yeah. Okay.
So for anaerobic respiration, that starts with glycolysis, so know that. And then it also ends with pyruvate getting converted into some other compound. And that happens because NADH is not called away by oxygen. So the NADH uses its its electron to convert pyruvate to something else. Okay, so that's why this happens, that's how this happens. NADH has no oxygen calling it away, so instead it reduces the pyruvate and NAD plus is regenerated, and then we have some other organic product. You only get 2 ATPs total, and those are from glycolysis, not even from the fermentation process. Okay, but the whole point of anaerobic respiration is to regenerate that NAD plus, so that glycolysis can happen again, because NAD plus is a limiting reagent. We don't have like unlimited NAD plus. Okay, so then for aerobic respiration, I want you to be able to recognize when I say glucose is oxidized to pyruvate, that means I'm talking about the process of glycolysis. Okay, so glucose is oxidized to pyruvate, yep, that's glycolysis. I want you to be able to have a checklist and say all of these things happen in pyruvate oxidation. So I'll have a list of different things that are going on, and you have to tell me, yep, that happens during pyruvate oxidation, or no, that doesn't happen during pyruvate oxidation. So pyruvate oxidation, remember pyruvate is oxidized, so that means it's broken into acetyl and carbon dioxide. That also results in reduction of NAD plus to NADH. And then the acetyl combines with coenzyme A. Just remember, no ATP gets made during pyruvate oxidation. And then for the Krebs cycle, all I really need you to do is explain this in a couple sentences. Okay, so you don't need the deep details. So the acetyl-CoA binds with the starting molecule to make citric acid. That gets oxidized, and carbon dioxides break off. Also, the electron carriers are reduced. So NADH and FADH2 are made, as well as a little bit of ATP. So products of this are carbon dioxide, ATP, NADH, FADH2. I won't give you this diagram, so this should be from your notes, okay? So just a couple sentences
Can you go back one?
summarizing that you've got oxidation happening to produce the carbon dioxides. There's also reduction of the electron carriers and ATP produced. That's pretty straightforward. So that's you don't have to know all the numbers of different ATP and NADH and FADH2, just the products and how that was made. Okay, just a little summary. The thing to put in detail though is describing the electron transport chain. So you want to make sure that you talk about the electron carriers bringing their electrons to the proteins embedded in the membrane. The electrons move from protein to protein until they reach oxygen, which then forms water. As they're moving protein to protein, they're pumping hydrogens into the periplasmic space outside the cell. And so that is done by active transport, pumping. And then the protons move from high concentration to low concentration through ATP synthase, that's chemiosmosis. And that drives the phosphorylation of ADP to ATP. Okay, so just that, I'll give you this labeled diagram, and then you just explain it in words.
So you're gonna put that, and then we have to explain what is it?
That's right. You have that, and I have words like make sure you use NADH, electron, proton, active transport, chemiosmosis, ATP synthase, water, oxygen. Should be it.
So one, start with NAD,
So
go
I'll give you the
to-
words, give you the picture, and you put it together for me. Okay? So I know it's a lot for 2 hours. I think it'll be manageable.
How many
if you
questions
prepare.
is it?
There's 15, like, look at stuff questions. And then there's the short answer, and then there's the 2 longer ones, which is the electron transport chain and describing the experiment.
Okay.
And then the rest are like short answer. Okay? You guys gonna be ready on Wednesday?
No,
Yes. Good. Remember, there are